Bismillahirrahmanirrahim. So we come towards the chapter number 4, that is actually the last chapter of the foundation module. Thankfully, you made it to the last chapter, and now we are going to do it very quickly so you can learn each and everything. I've always told you that there is extracellular fluid and intracellular fluid. The extracellular fluid is the only statement to remember the ions is cell is a bag of potassium. So inside the cell, we are going to have mainly potassium ions. Then there will be proteins and phosphates. And in the extracellular fluid, the main ion will be the sodium ion. This is that. And some can be chloride ions, etc. Now we have this lipid bilayer over here. So this lipid bilayer shows us a lot about the structure of the cell. The cell membrane is a lipid bilayer. Phospholipids are there, cholesterol is there, and proteins are there. Protein make 55%, phospholipids make 25%. So the substances which can easily move through the cell membrane, they undergo simple diffusion. These include oxygen, carbon dioxide, and alcohol, etc. The substances which cannot easily pass, they are provided to pass through the membrane by the proteins. The proteins can provide a channel for the molecules to pass through, and these are known as channel proteins. Here, a channel is present through which a molecule can either move inside the cell or move outside the cell without requiring any energy. There can be facilitated diffusion. This is also a type of diffusion, and this substances can move in the cell or out of the cell without the usage of energy. But in this case, the protein, which are the carrier protein, they undergo a conformational change in which a certain molecule it attaches onto certain substance. The carrier protein undergoes certain conformational change and brings the molecule to inside or outside, opposite of which. But in certain cases, the carrier proteins, they require energy in the form of ATP to carry the molecules either inside or outside of the cell membrane, and that is known as the active transport. So we have diffusion, which can be simply moving inside or outside, moving through the channel proteins, moving via carrier protein, that is facilitated diffusion, or it can be active transport. This is the main crux of this chapter. And the protein molecules that have their portions moving through the whole of the cell membrane, they are known as integral protein, whereas the ones which are located on the outside or on the inside only, they are known as peripheral proteins. So here we have a mini comparison between diffusion and active transport. Diffusion is the movement from an area of high solute concentration to an area of lower solute concentration, or by the random molecular movement of the substances, and this is due to the energy of the normal kinetic motion of the matter. Matter is continuously in motion, and due to this normal kinetic motion, the process of diffusion takes place, whereas active transport requires energy, which is in the form of ATP, and ATP is produced by mitochondria from the food particles we studied in the chapter of organelles. So the continual movement of the molecules among one another in liquids or gases is diffusion, and it is due to the normal kinetic energy of motion of the molecules. And diffusion can be of further two types: the simple diffusion, in which the molecules just move across the cell membrane without even a protein molecule. But sometimes proteins are also involved in simple diffusion process in the form of channel proteins. But when carrier proteins are involved, the diffusion becomes facilitated diffusion. This diffusion depends upon the number and size of openings in the membrane through which the substances can move in or out, the velocity of kinetic motion, that is the normal constant motion, and the amount of the substance available. Normally, diffusion occurs from an area of less concentration, from an area of higher concentration to an area of lower concentration. So it depends on amount, number and size, and velocity or the kinetic motion of the molecules. Again, they have mentioned simple diffusion occurs through the lipid bilayer. This is for the lipid soluble substances like oxygen, carbon dioxide, and alcohol, but it can occur through watery channel proteins. These channel proteins provide a passageway for the transportation into or out of the cell. Again, is of the lipid soluble ones oxygen, nitrogen, carbon dioxide, and alcohols, which can pass the lipid bilayer easily because they are non-polar and they can easily be soluble in the lipid bilayer. Their rate of diffusion is directly proportional to their lipid solubility. Now, the pores that were provided by the proteins that were channel proteins, these protein pores are known as aquaporins, and there are 13 different types of aquaporins in animals. And if we take the example of the red blood cell, the movement of the molecules via the channel protein is 100 times as great as the volume of the red blood cells itself. And this movement through these channel proteins is 1,000 times as great as compared to the movement of the molecules in water. Here we talk about the protein channels, the aquaporins actually, and the movement of the substances through the aquaporins, it depends upon the diameter of the pore and the electrical charges of the pore. So depending of which we get the selective permeability of the pore, and the pores can be either voltage gated or ligand gated. Voltage gated is that they require a proper voltage for their opening, whereas ligand gated means they require proper substances to be attached onto the receptors for the opening of the pores, protein pores. Now we will have a look at the protein channel, that is the potassium channel. But before that, we know that protein channels are highly selective, and the movement of the molecules across them depends upon the diameter, shape, and nature of the electrical charge that is present on the molecule to be transported. Here we look at the potassium channel. So this potassium channel actually is composed of 4 pore helices, on the top of which there are 4 pore loops, and on the pore loops we have a selectivity filter. This selectivity filter is actually composed of carbonyl oxygens. When the hydrated potassium ions they come over here, they interact with the carbonyl oxygens. These potassium ions they lose their hydration, they become dehydrated, and in this way they can move from the outside to the inside. This is the method of movement of the potassium molecules from the outside, in which they are hydrated, to the inside where through which they become dehydrated. But now the sodium ions they cannot move through the same potassium channel pore. This is because the sodium ions they are very much small and they cannot interact with the carbonyl oxygens, and hence they cannot pass through the potassium channel. Now we have the sodium channels. The sodium channels they are 0.3 to 0.5 nanometers in diameter, and the interior of these channels is negatively charged and their gate is closed. Whenever their gate opens, the sodium they are attracted towards the interior through this negative charge, and they come to the interior, and then the gate is again closed. This is depending upon the voltage. So these are voltage gated sodium channels. Since sodium are more in number on the outside, so they move from the outside to the inside, or from higher concentration to the lower concentration. So net movement is from outside to inside. For potassium, their main gate is on the interior, and potassium ions are more on the inside, so they need to move outside. These are the potassium channels, and the potassium gates open. They are also voltage gated channels, and this voltage is actually very important in the transmission of the muscle and the nerve fiber impulses. So when potassium ions they move out to the gate, from the interior to the exterior, from their higher concentration to their lower concentration, this allows repolarization to take place. Again, they're talking about the gating of the protein channels. The protein channels can be voltage gated by the voltage, which is mainly the gating of the sodium channels and the potassium channels, but sometimes the protein channel can be gated via the ligands. In such a situation, the acetylcholine receptors are present, and such channels are known as acetylcholine mediated channels. So consider this diagram. For example, this is an area of acetylcholine receptor. So whenever an acetylcholine molecule will attach to the receptor, it will cause the opening of this protein pore, and hence a channel will be created. This is a acetylcholine gated channel, and such a channeling is known as ligand gated ion channeling. Now when the acetylcholine is attached, it allows the opening of a 0.65 nanometer negatively charged channel, and hence the sodium ions they can move through this voltage to this ligand gated channel to the interior of the cell. This is very much important for the transmission of nerve signal from one cell to another. We will study this in detail in the nerve physiology chapter. These gates, they have open state or closed state. They have no intermediate state. They either open at one voltage or they close at other voltage. There is no hemi opening or partial opening of the gates or channels, and the opening only lasts for a fraction of a millisecond, and then again they close after that with a snapping, and there is no partial opening and partial closing. This is known as all or none fashion of the opening and closing of the gated channels. So now we have a very nice method to actually measure the properties of certain ion channels. This method is known as the patch clamp method. In this method, what we do is we take a certain pipette and we take a very small area of the cell membrane. And this way, we get a very small patch which is clamped by this pipette, and this membrane patch is entered, and this membrane patch is so small that it contains only single protein channel, and it is put into a solution of either the ion that is to be tested, and whenever the molecules move across the membrane, it is attached to a recorder, and we can detect any movement across this, and this is how we can get the properties of the channel, either they are voltage gated or either they are ligand gated, and which type of proteins or substances or ions that they are allowing to be moved through the membrane.